Lastly, a diagram or a video clip has to be provided for this process so that they can further understand what the whole process is going to be. The next topic that is going to be discussed under Chemistry of Life is the process of protein synthesis, or more known as the process of transcription and translation. So under this topic, there are several things that are needed to be discussed because they entail processes that is needed to be written down in most essay exams. So actually, apart from DNA replication, this is also one of the topics that is commonly being asked in essay. So what are the things that is needed to be discussed? So first, the significance of the topic. Of course, the significance of the topic is of the process of transcription and translation is to produce the proteins according to the codes, or according to the information that is being kept in the DNA. So all the processes that is being undergone by the cell is being dictated by the DNA of the cell. Okay, so right after explaining the significance, we can now move on to explaining the nucleic acids. So I think I have included this in the previous chapter, that in explaining the structure of DNA, the nucleic acids has to be discussed first, because nucleic acids, they make up the genetic information molecules, which are all made up of nucleotides. So same thing, in the process of transcription and translation, it also entails the use of the DNA as well. So the structure, the familiarity of the structure of the DNA has to be laid down in this area as well. So in the nucleic acids, we have to introduce the other type of nucleic acid, which is the RNA. Okay, so the most common things that are being asked according to this topic is the comparison between the structures of DNA and RNA. Between DNA and RNA. So you have to write down the features that we can compare DNA and RNA from. So for example, according to structure, DNA is double-stranded, and the structure of the DNA is in double helix, whereas for RNA it's just usually single-stranded. The nitrogenous bases is also being compared here, where in DNA uses thymine, the nitrogenous base thymine, while RNA uses uracil. Okay, so writing down the features, okay, would help the students compare the two structures. Okay, so after comparing the two nucleic acids, we can now continue with the different functional types of RNA. So in the case of functional types of RNA, we can introduce to the students that different types of RNA is being used in the process of transcription and translation, such as the mRNA, the messenger RNA, the transfer RNA, and the ribosomes. So we have the mRNA here, the transfer RNA, and the ribosome. Okay, so under the functional types of RNA, we have to include the structure, okay, and the function of each type, or where they're being used within the process of transcription and translation. So the next type, so right after explaining the structure of nucleic acids and highlighting on the area of RNA, we can now move on to the process of protein synthesis, or the transcription and translation part. Okay, so the students might be puzzled, what is protein synthesis? Protein synthesis is also transcription and translation. It's just that both processes encompasses this. Okay, so or is being encompassed by this. So we have to note that the protein synthesis, we are able to form the proteins because we are able to use the process of complementary base pairing as well. Just the same as the ones that is being used in DNA replication. So so in the process of transcription, this is the formation of mRNA. So basically, it is a complementary copy of the DNA. So we are only able to make a copy of the DNA through the process of complementary base pairing. Okay. So right after the formation of mRNA, okay, so this mRNA will now be used in the next process, which is the translation. So this one, okay, we are now going to basically translate the information that we have from the mRNA and then translate it into a protein using another RNA, which is the tRNA. So into a polypeptide, which is the primary structure of protein, using tRNA. Okay, so by giving a general idea of what the process is, at least the student will have an idea of we are actually giving an expectation to the students that this is going to be the processes. Okay, so under this process, okay, so we have to include the location where the protein synthesis is occurring. So we have to indicate that in transcription can only happen in the nucleus, wherein it uses the mRNA. And translation occurs in the cytoplasm, or more specifically in the ribosome in the cytoplasm, where the formation of polypeptide occurs. Okay? So after including the location, we have to include the enzymes that are being used. Okay, so by including enzymes, then by knowing the enzymes, then therefore we are able to write down what their functions are. Okay, so in the case of transcription, it only uses RNA polymerase. So in the case of RNA polymerase, it is able to form the mRNA molecule through the process of complementary base pairing. So another one, okay, although in translation it doesn't really use any enzymes, but it only uses tRNA and and the three-dimensional ribosome. Okay, and then translated into a polypeptide. Okay, so it'll be more helpful as well if we are going to provide diagrams for the students for them to be able to see what the process is. So, having written all this, okay, the students might ask what codons are. Okay, so along the process, along the formation of the mRNA, okay, we have to include the genetic code. Okay, so the genetic code, it is actually a sequence of bases. Okay, so they're actually made up of codons or triplet codes that is complementary to an amino acid. So let's just say in the let's just say the codon UGA. Okay, the codon UGA is complementary to methionine. Okay, so if this codon UGA is present in the mRNA, then therefore later on, when it's going to be translated in the process of translation, then the amino acid that is going to be assembled first is the amino acid methionine, which is going to be the first amino acid in the chain of the polypeptide. Okay, so there. Okay, so last but not the least, okay, the last thing that is going to be discussed under transcription and translation is the one gene, one polypeptide hypothesis. Okay, since the DNA holds several hundreds and thousands of bases, it can also contain several genes. And those genes will code for a specific polypeptide or a specific protein. Okay, so under this hypothesis, okay, so the gene comprises specific sequences of bases that will code for the assembly of a specific polypeptide or a specific protein that is going to be produced. Okay. Sequences of bases. Specific polypeptide. Okay, so this process, okay, so the process of transcription and translation under the chapter Chemistry of Life is basically a gist or just a summary of what is going to be discussed later on in chapter 7, which is nucleic acids and proteins. So this can be applicable in both SL and HL students.